So this is prep 3 for lecture 20, not 21. We are going to just quickly review. So the F factor is a plasmid. It is a small circle of DNA that is somewhat independent of the bacterial chromosome, so it can replicate on its own. And when bacteria conjugate, it is the F factor, the plasmid, that gets transferred. It has all the genes on it for making the pilus, which is the structure, the little tube that allows the transfer of the DNA from one cell to the other. And it has all the genes for the actual transfer part as well. And so most of the time, the F factor just stays as a separate DNA. And it's actually the relative sizes of the bacterial chromosome and this plasmid are grossly underestimated. So the plasmid is much, much, much smaller than the bacterial chromosome. But for the purposes of these schematics, we need to show things a little bit bigger than they really are. But there's an occasional rare event. There are some sequences around the bacterial chromosome, and the F factor has a set of these as well. And it means that sometimes there can be crossover between the plasmid and the bacterial chromosome, and that will cause it to integrate into the bacterial chromosome. So if you have a circular chromosome and then you have a circular plasmid, if you have a single crossover between them, a breaking and rejoining, I'm just going to change color so you can sort of trace it. So what will happen is you will get, here's your big bacterial chromosome, and then it's been broken, and so it connects now to the plasmid, and we trace the plasmid, and then this end has been broken and connected. So what we really end up with is one a little bit larger circle, but it's got a small amount of the plasmid, and then the rest of it is the bacterial chromosome. And that's what's shown here, in no way to scale, because the plasmid's very tiny. But what happens now is that this factor, which promotes its own replication and transfer into a recipient cell, is now part of a really huge circle of DNA, and that whole entire chromosome acts like a gigantic F factor. And so it's referred to as high frequency recombination strain, because this is a case where you can actually see transfer of the chromosomal genes, the bacterial chromosomal genes, from a donor cell into a recipient, instead of just the DNA that's on plasmids. And there's a few strains like this. There's a number of different ones that are available to researchers who work on this stuff. And so this just shows an example. These are these insertion sequence elements. And if you were to look around the bacterial chromosome, you'd find that there were these IS elements kind of in different places all around the chromosome. And a plasmid has, the F factor has found or picked up or somehow acquired a sequence that's similar to that. And when crossover occurs between two pieces of DNA, it does so because of sequence similarity. So on eukaryotic chromosomes, there's crossover between the chromatids of a pair of homologs because the DNA sequence is almost exactly the same. And it is that sequence similarity that allows them to line up the way they do and then cross over. And that's the same for bacteria. The sequence is the same for the IS elements, and so therefore they can sort of find and pair with each other, and if there's some breaking and rejoining, we can end up with this is not a linear DNA. Let's just show that it's part of a circle, right? So this is going to curve around at some point, but it's a circle that now there are two copies of that IS sequence, and you've got your plasmid inserted in between. And then when the HFR cell takes part in a conjugation, now instead of just the plasmid being transferred, the bacterial chromosome gets transferred. And this is the same mechanism. So there's a break in the DNA, and a single strand of DNA is fed into the recipient cell. And then as that single strand is fed in, DNA polymerase 3 is busily making this double-stranded again, and when it reaches all the way around, if it did, it would be complete. The truth is that this process would take a really long time. It takes like over an hour and a half to transfer that whole bacterial chromosome into a recipient cell. And so most of the time the pilus destabilizes or breaks before that. So it's very, very unlikely that this whole piece of DNA would get incorporated into the recipient cell. And even if it did, the genes that get incorporated into the host's bacterial chromosome are going to have to do so by crossing over. And so it's almost impossible. It's not impossible, but it's close to impossible that the F factor is going to be transferred and then integrated into the recipient cell. Let's see, is that everything I wanted to explain? It's replicative transfer, just like the same with the plasmid, in that a single strand is fed in, and then the host makes it double-stranded as it's coming in. And the single strand that's left behind here is basically made double-stranded again. So whatever genes are actually transferred into the recipient cell are still retained. They're not lost from the donor. Okay, but one key difference between the kind of conjugation that involves the transfer of a plasmid is that these cells remain recipients. They don't become donors because they don't generally receive the F factor. It's just a peculiarity of the way the transfer of the DNA takes place, is that if you were going to do it the full thing, it's actually the part of the F plasmid is right at the tail end. So this is this is where all the genes for making a pilus and regulating the replicative method of getting the DNA into the recipient cell is all encoded. And so this cell, it may change its genotype with regard to some of the other genes, but the F factor kind of stays in the donor cell only, and the recipient cell remains a recipient cell. Okay, and so basically you can't just transfer a bunch of linear DNA into a bacterial host cell because there are nuclease enzymes that do not like single-stranded DNA. If your chromosomes are supposed to be circular, then when single-stranded, or not single-stranded, sorry, linear DNA that's not in a circle but has these ends on it, when those are recognized, those could be certain kinds of viruses invading the cell. And so there are protective enzymes that just gobble up any kind of linear DNA that is introduced into the cell. And so there's a limited amount of time in which you can incorporate some of the donor genes into the recipient cell. And that requires crossing over. So this is the only kind of case where you have a very short time. You're sort of a partial diploid because there are in fact two copies of certain genes in the recipient and the donor. And it requires an even number of crossovers because if you make a single crossover between a linear piece of DNA and a circular piece of DNA, whoops, we should go this way, then what we'll end up with is a linear piece of DNA that will get chewed to bits. You have to have two crossovers in order to complete the circle. So let's just show that again. I'm gonna, maybe red is a better color. So if we just had this one crossover, we'd have this linear molecule. And then when we cross over, we've still got this linear molecule. So instead what you need is you need to cross over this way and then crossover here, and then that incorporates this bit of DNA into this circle. The little bits of linear DNA that are left over, because there will be some that have some of the host chromosome and the donor chromosome in them, that part gets chewed away. So you're only going to have, at the end of the process, you're only going to have just the circular chromosome. It's the only part that can be stably maintained in the cell, but it now may have a change in genotype. It depends on what the genes were, here. If the recipient has the wild type A and the donor also has the wild type A gene, then even if a crossovers occur, you're not going to see a difference, right? But if this was actually A minus, so unable to make its own whatever A is, and this was A plus, then we would see a change in genotype. And part of the way, one of the most important aspects really of experimental design with bacteria is the way you design the genotypes of your crosses. Okay, and so there's a couple of names to just know. So here's our HFR strain and our F minus strain. During the time, so notice this is a single strand of DNA coming in, but then it gets turned into a double helix once it's inside. And this is called an X conjugate. X conjugate is just the term that's used for a bacterium that's taken in some donor DNA and that has its own bacterial chromosome. So I don't think these terms are that useful, but endogenote or endogenote is the bacterial chromosome, and the exogenote, that's the DNA that's been introduced that came from the outside. So endo, inside, exo is from an external source. It came from elsewhere. And then the recombinant, so there was crossovers, one here and one here, and in this, just for this one cell, that allowed these genes to be taken in onto the bacterial chromosome, and the genes that it had, the A minus, B minus, to actually be exchanged. And then this gets chewed to bits, and you end up with a bacterial cell that has had two crossovers and has taken in these two genes and acquired them. And if we were to plate it on a media that lacks the A and the B, it would be able to still survive. The original recipient cell couldn't, but the crossover, the recombinant cell could. And it is referred to as a recombinant once, once this process has completed. Okay, and so this is just a picture. This is just a reminder. The F factor incorporates into the bacterial chromosome, and it creates an HFR strain. The F factor is indicated this way. And the little arrowhead is really important when you see this symbol, because it means that this is the DNA that's going to be cut and fed into the recipient cell first. And so all of these genes are going to be transferred before this part here. This is the part that's required to be a donor, and it is transferred last. Now, the transfer of the whole entire bacterial chromosome on average, it's usually around the same size, and it takes about 100 minutes, so a little over an hour and a half to transfer. And in the kind of work that is done, we don't actually let the experiment proceed for that long. So the F factor is almost never even introduced into a bacterial recipient cell. But even under natural circumstances, if you were to let the mating proceed, it's the pilus doesn't maintain stability for that long, because it's normally a structure that's only needed for like a 20-minute transfer of like a plasmid. If 20 minutes. And so it's not meant to actually transfer this huge long chromosome, and so it doesn't usually stay stable for long enough. The connection between the cells breaks. And we can use this process of conjugation with HFR strains to get a crude map of approximately where different genes are relative to one another by doing what is called interrupted mating. So what you do is you take an HFR, culture of HFR cells of known genotype, and then recipient of the correct genotype, and we'll talk in the next lecture about how you choose those genotypes. And you mix a large amount of them together, and then you allow them to mate, and then every minute you you kind of disturb them by putting them into a blender and turning the blender on. The bacterial cells do not continue to mate under this condition. And so after, and you only have to blend them for 3 seconds or something like that. And then you take that aliquot and you plate them out to find out what the genotypes are. And you can determine, usually we're trying to map genes that are sort of in this region, and we want to know, well, which of these genes comes in first? Is it the adenine gene, or is it the histidine gene, or is it actually the lac genes? Which one comes in next? Which ones show up close to each other in timing versus which ones are delayed quite a bit? So the idea is you can use the time. If you stop the process every few minutes, then over the time, more, the longer you wait, the more of that bacterial chromosome was filled in, and therefore there's more chance for some of these genes that are further down the chromosome to actually get into the bacterial cell and then be incorporated into that bacterial chromosome. So interrupted mating is a way of generating the beginnings of a map. You would then do some more high precision mapping later once you had a sense of which genes were located where. And I think that this will be enough. I think we will start here for lecture 21. So just take a look at this slide and just think about this. Careful design of different genotypes in the donor strain and the recipient strain are very, very important, because what you don't want is to culture your mixture of cells and be getting most of the bacteria that grow are just your F- strain, and they haven't actually participated in mating or taken up any DNA from the donor strain, or they're a bunch of donor cells that are just able to make their own whatever nutrients. So maybe take a look at this and just read through this, and then in the next lecture we will start here.